Module 2. And we will start with the biological membranes and the transport of the small molecule. And so first, we are looking at the plasma membrane, the phospholipid bilayer, and the functions. These are the very generalized, all straightforward. So this is a picture, and take a look at this picture and spend some time on that. Because see that I have written or highlighted, observe this and similar pictures on the internet to have a concept of what is what. So when you look at the pictures, you will see some of those pictures in your exam, and you have to make conclusions out of that. Say, for example, these are all the whitish or the grayish parts are the phospholipids. So you have the phosphate head group right here as a round, which is hydrophilic, that is facing towards the exterior or the cell environment. Do have two fatty acid tails. These are hydrophobic. And on the cytosolic side, this is right here is a cytosol. So you can see right here, cytoskeleton, cytosol. Those are also hydrophilic facing the cytosol, which is aqueous. And you have two fatty acid tails sticking in towards the middle part. And that is hydrophobic. So this you have a hydrophobic core and the hydrophilic head groups, one of them on the exterior and one of them in the interior. So what we call that outer part, this layer, as the exoplasmic phase, or which is facing the exterior, exoplasmic phase. On the other hand, this leaflet, which is half of it, of the phospholipid, we call this as the cytoplasmic or cytosolic, both interchangeably, cytosol or cytoplasm, same thing, is the cytosolic or cytoplasmic phase. So these are the two different phases of those, because if you just cut the lipid bilayer into the outer half of the layer, our inner half of the layer, that's how you will name it. And you can expect some of the questions that we actually do ask, that which of the following architecture of the biological membrane is true. So it is basically a hydrophilic layer, hydrophilic head groups, followed by an hydrophobic core, followed by another hydrophobic fatty acid layer, then followed by a hydrophilic layer. So if you just mentioning those, so hydrophilic, then hydrophobic, then again hydrophobic and hydrophilic, if you're going from one side to the other. So just learn those, that if we just describe the phospholipid bilayer, where is what, which we called as the membrane topology, that the topological arrangement of the membrane and where is what. So that is very important. So definitely you are going to expect questions based on the topology of the membrane. Where is what? Exoplasmic phase, or the exterior phase, is facing the phospholipid head group, which are hydrophilic, followed by those have the fatty acid tails, which are buried in the lipid bilayer. Then you have another layer of fatty acid tails buried in the bilayer, followed by hydrophilic head groups, which are sticking towards the cytosolic or the cytoplasmic surface. So I just repeated it twice, because it's really super important. All right. So after that, we are looking at many different proteins. So the proteins, there are several different types. So first of all, when we are looking at this protein right here, like a channel, this is like a blob, and these are all spanning the entire membrane. So you can tell them they are transmembrane protein, and AKA also known as the integral protein. Now, some of the cell biologists at the very higher level sometimes distinguish in between integral and transmembrane proteins, but usually we will not go into that detail of those. So anything that you see is sticking out towards the exterior, sticking out towards the cytosol and continuous through the membrane, you will consider them as an integral protein, transmembrane protein. Both the words, we will use them as the similar name. Now, here, there are a couple of proteins buried in here. And if we are looking at these proteins, we do not know or see that whether they have gone through the membrane completely. So we will not know. So from here, I will assume that because I cannot see anything, I will assume that they are peripheral proteins. But again, you will not see something like this, which are ambiguous. Like they are not, you cannot visualize the rest part of the protein, which is buried in the membrane. You will be able to see proteins just like what you have in the front. Now, there is another type of protein, which will be the peripheral proteins. So the peripheral proteins, and this is the important part. You see that there are two peripheral proteins which are shown here. One is right here, one is right here. And again, remember that they could be either side. And both of these protein are associated with another protein. So you have one transmembrane protein. Here you have usually 1, 2, and 3 transmembrane protein. You have one transmembrane protein, I am assuming, and this is a peripheral protein associated with that. Now, there is also another picture. We'll see those pictures as well. Peripheral proteins can be associated directly onto the plasma membrane like this, partially buried right here. So they can be attached with a protein, another protein. So this portion is the protein as well, or they can be directly buried into the plasma membrane, which that is not been shown in here. So if you see a protein partially go through, this is just partial part, not the entire part like this, or all of these span the entire membrane. These are all integral membrane proteins. And this is partial. That's why this will be as a peripheral membrane protein. Very important. Other than that, you will have a very important clue here. And this, you should take extra importance to this one. So there is a protein which is called a lipid anchored protein. So you should see a protein like this, a blob here. Underneath that protein, you are going to see there will be 2 fatty acid tails, just as you see in green. If you see that there is a single protein which does have these directly fatty acid tail, and of course with a similar different color. I mean, like, I will just not draw a gray colored picture in here. If I just draw that, you may think that this is like a peripheral protein. Like, this is, you cannot distinguish that whether this gray protein is a peripheral or lipid anchored. But when we will give you a picture of the lipid anchored protein, that will be different color than the rest of it. So see, this entire part is green, which is meaning that this entire entity is just one entity. And this is your lipid anchored protein. Why it's a lipid anchored? Because these are the fatty acid tails. That is an integral part of the protein. So this is also known as a lipoprotein because it's a lipid plus protein. It's of course a lipoprotein. Along with that, it is also what is called a lipid anchored protein. So just make sure that you know this very, very well. You will see pictures. You will see pictures in your exam. Suppose you see a protein like this, and if there are 2 tails, all you have to see that look for tails. Look for tails. So that is your clue, that if you see that there is a protein on the surface with the tails, they are your lipid anchored proteins. All right, so that's pretty much it. So let's go into a little bit of detail about that. Depending upon that, how important the material is, I will spend some more and less time depending upon what it is. So here, just to know that the lipid is what we called as an amphiphilic. Similar to the amphiphilic, you also see a word which is amphipathic, and both of them can be interchangeably used. Amphipathic. What we mean by this, amphi means both. So that means the amphiphilic means that it does have a hydrophilic end plus an hydrophobic end. And again, an amphipathic means that it does have a hydrophobic end and a hydrophilic end. So basically hydrophilic and hydrophobic. Any of those two, if you have that, you can call them amphiphilic as well as amphipathic. This can be interchangeably used. So here, what you're seeing, there's a head group. This is polar, what means that it contains charges and it can form hydrogen bonds. And these are the fatty acid tails. And 3 classes of lipids: phospholipids, sphingolipids, and the sterols. Know those, very straightforward. So let's move on to some of the important part. So here we are looking at from the 3 different classes: phospholipids, sphingolipids, and the sterols. We are looking at the first one, which is the phospholipid. So the phospholipid is a generic name, and there are many different types of phospholipid. And here we have one example of that. So out of all phospholipid, the general structure is that they will have a phosphate head group associated with an glycerol. So you can see that PO4 minus, POOO minus, PO4 minus. And then this is, you have a CH2OH, CH2OH, and CH2OH. That is a glycerol structure. And that hydroxyl end of the OH is bonded with the phosphate. And the other two hydroxyl OH is bonded to the carbon fatty acid tail. So this glycerol molecule is in there as well. And then you have two fatty acid tails. So how there are just basic structure, but you have literally, there are hundreds of different phospholipids, and how these phospholipids actually vary in their structure, in their quality, is essentially, just think about that you have a phosphate, you have a glycerol, and if you have a choline right here, this one right here, and this will be known as a phosphatidylcholine. That is one of the phospholipid. Now it contain, this is one of the fatty acid and one of the fatty acid. Now, the diversity of these fatty acids can vary. Suppose I'm just saying, just randomly, that you have a lipid and you have a palmitic acid in here, and you can have a butyric acid. And depending upon that, say, for an example, if you just extract the coconut fat, where you will have a lot of palmitic acid. The name palmitic comes from the palm trees, coconut-related fatty acid. On the other hand, if you just take a look at the buttery fat, those fats contain actually butyric acid, and those butyric acids are the fatty acids which will make up the tails. So the tail composition will vary according to the different ones, and depending upon the composition of the lipid, these different fatty acid gives the different texture and taste, and the room temperature liquid conditions, like say, for example, canola oil, vegetable oil, and all these cooking oils, they are liquid. But say lard or some coconut fat, these are solid, or butter, they are solid at room temperature. So depending upon those composition of these fatty acid tails, their characters actually vary. So we are going to take a look at what are these characters. So one of the characters of these fatty acids, that they may be containing all single bonds in between their hydrocarbon tail. So these are hydrocarbon, CH2, 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 and see that if you look at the left side of the fatty acid, all of them are bonded with a single bond. This is like a single bond, single bond, single bond. And these are very straight. These are straight. They do not kink or bend. And they are actually much more stable in temperature variation. On the other hand, what happens and these are called the saturated fatty acids. Saturated fatty acid means that all of them are bonded. So if you can look at that the carbon actually have a valency of 4. So you have a 1, 2, and then connected to 2 hydrogens, means it's a 4. So all the 4 valencies of the carbon is occupied. And that's why we called that they are all saturated, a single bond. So on the other hand, the other type is an unsaturation, where you have a single bond in between the 2 hydrocarbons. See that you have a CH and the CH. So where are the 4? So you have the carbon, you have a 1 bond here, carbon to hydrogen, 1 bond. Then you have a 2 bond. So 1, 2, 3, and 4. So this is an unsaturation, which is a double bond. So if you have a double bond in a fatty acid, we call them as an unsaturation or an unsaturated fatty acid. In a saturated fatty acid, every bond is a single bond, whereas in an unsaturated fatty acid, it is not. There is usually 1, 2, 3. Usually that is the maximum number. So there is not a fatty acid which does have an infinite number like 10, 12 double bonds. That is not seen. Usually they are 1, 2, or 3 unsaturations only. And what are the different characteristics? That is important. We are going to talk about that a lot. If you have a double bond anywhere on these fatty acid tail, form a kink right here. This is basically, see that this is straight. This is a kink in here. And that packs the fatty acid tail in a different way. And depending upon the temperature, they tend to be much more liquidish towards the room temperature. And again, do not quote me on this. This is at this point, I'm just introducing that term, and we will go into the detail of the molecular detail of that pretty soon. But happens that if you just think about the melting temperature of the fatty acid, the melting temperature of the fatty acid with unsaturation will be much less compared to ones which are with the saturated fatty acid. Just think about that this way, that say, for example, if you take the animal fat or lard, which is solid at a room temperature, they will tend to have more saturated fatty acids, which does have all single bonds. Whereas if you take a canola oil or olive oil, they will tend to have more of an unsaturated fatty acids in their content, which will give them this property as well. Now, remember that when we are talking about all these oils or fats, they are composed of these fatty acids as well. The composition here, these fats are concentrated on the plasma membrane of the cell. But on the other hand, say, for example, when we isolate the animal fats from a source, some of the sources of these fats are coming from the lipids of the cells that are made up of. There are some specific areas where you have extra adipose tissue where lipid actually accumulates inside the cells, called the adipocytes. But all the fats are made up of these same things. So they are not separate, two different things. So after that, we are looking at these unsaturated fatty acid with a kink double band and the saturated fatty acid. The two phospholipids can have so again, this is already I mentioned, have the same head group, but may have different fatty acid composition of the carbon chain length or degree of unsaturation. That's how phospholipids generate a huge diversity in membrane composition. So this is what you are seeing, that this is an oleate, or this is an ionized form of the oleic acid, whereas this is a palmitate, ionized form of the palmitic acid. So choline right here, phosphate glycerol, and you can have this one as oleic acid, this one as palmitic acid. So this can be oleic acid, this could be palmitic acid, and that will be one kind of fat. And then you change this one, that one could be butyric acid, another could be oleic acid. And then you keep this one, that one will be palmitic acid and another will be something like a linolenic acid. So you can make permutation and combination of the fatty acid tails with the phospholipid head groups, and that will generate all these diversities of the fat. So the main types of fatty acids or the associated lipids that are present in here are these four types. These are the most abundant on the plasma membrane. So again, you can see that at the bottom, we just only wrote fatty acid tail. And why we wrote fatty acid chain or tail? Because they can vary. What is constant is the head group. So let's take a look at one at a time. So here you have, again, a phosphate, CH2CH2 right here. This is the glycerol part. This is the phosphate. And you have a CH2CH2 NH3 plus, which is ionized. So see that right here, this is a plus charged. Here you have a minus charge. So they neutralize each other. And this is known as a phosphatidyl ethanolamine. So this is an NH3 plus is the ionized form. So this is a zwitterion. So if it's an NH2, CH2, and those will be an amine. So that's how this is an amine. So phosphatidyl ethanolamine. So that's one of the type. This is a neutral lipid because you have a plus and a minus, and this zwitterion cancel each other. The next one, on the other hand, you have a phosphatidylserine right here, where you have, again, the glycerol, you have the phosphate, but see that right here, you have an NH3 plus, then you have a CHCO minus, CH2O, and the PO4 will always have a minus head group. So you have a one extra CO. So this used to be like in a non-ionic form. Remember that in a non-ionic form, this will be NH2, CH, COOH, CH2OH. That will be the serine. That is your amino acid. When it is bonded and in the ionic state, that serine amino acid, this is just an amino acid, has now in the ionized form NH3 plus, CO, the H plus is ionized, it's gone right here, and it is COO minus. So now you have a net extra minus charge. So phosphatidylserine, this is a negatively charged phospholipid. So this is a very important one and contributes to the membrane architecture a lot, phosphatidylserine. Then we are going to another neutral lipid right here. This is a phosphatidylcholine, which was just the same thing right here, just the molecular structure. You have right here CH3, you have an N, which originally it was NH, and this N is ionized, CH2OH, and this PO4 minus. So this plus and minus neutralize each other, and this is your phosphatidylcholine, and this is a neutral phospholipid as well. Next, you have another one. This is your sphingomyelin. The sphingomyelin, we are going to talk about that. It also an example of a sphingomyelin, which is a phospholipid. There are some of them sphingosine in this class of lipid, which could be a little bit different. So the sphingomyelin contains a phosphate group, and see that here you have a plus charge, you have a minus charge. They neutralize each other. So this will be also a neutral lipid. So out of all those, the only charged, negatively charged phospholipid is the phosphatidylserine. And other than that, phosphatidylethanolamine, phosphatidylcholine, sphingomyelin, all are neutral lipids. So just the general structure of that will be helpful. All right. So after that, we are looking at another kind of phospholipid, and these are called phosphoinoside. And this is the lipid form, and this form is a signaling molecule. We are going to talk about this a lot when we go into the signal transduction chapter. But how it is formed, that this is your phosphatidylinositol. So this inositol is a hexose ring, which is derived from a glucose. So this is a sugar. Remember that. This inositol is a sugar derived from the glucose associated with a phosphate. This is your glycerol, and you have a fatty acid tail. So compared to these, right here, this is upside down. So the head group is on this side. So this is again a sugar head group, phosphate, then you have the glycerol, 2 fatty acid tail. General structure is the same. And now what can happen to generate this molecule, there is a PI kinase which can phosphorylate another group of the inositol right here. The phosphatidylinositol becomes the PI4 phosphate. So you have what we call as a PIP. On the other hand, say for an example, the PIP kinase will phosphorylate it again, another position, and this will become a PI45 bisphosphate or PIP2. And this phosphatidylinositol is a signaling molecule. We are going to see that phospholipase C cleave that in 2 parts. This becomes a diacylglycerol. This becomes a IP3, inositol phosphate 3, which diffuse into the cytosol to signal endoplasmic reticular, different gated channels which are present on the endoplasmic reticulum. Not needed now. So the only thing our take home from here or from this slide will be which of the following can act as a signaling molecule. Just remember that these are one of the important. And also what we are going to see that at the later part, we can have another important thing that sometimes the lipid rafts we are going to talk about, they have high affinity for some of these particular proteins. We are going to talk about that later. So just a signaling molecule is good enough. All right. So after that, say, for an example, from here, phospholipids, we are going to the sphingolipids. So the sphingolipids has a head which is variable, and the tail is constant, which does have 2 fatty acid tails, as you can see right here. And the difference is that if the sphingolipid contains a phosphate right here, then it is considered as a phospholipid as well. So you can see that if this is the sphingomyelin, which does have the phosphate. So you can just think about that. Not all sphingolipids are the phospholipids. Remember that. That's the most important part. Not all sphingolipids are the phospholipids, but if they contain a phosphate in their head group, they can be considered as a phospholipid. On the other hand, what you do is that if you take this part out, take the part out and see that you have an oxygen and you bond this with this. So I take the not 2 oxygens, so 1 oxygen. Remember that. Just this oxygen directly bonds at this position. And now this is a glucocerebroside. So this is an hexose sugar again right here. So this is a sugar right here, a head group, and that's how the name comes in as glucosylcerebroside. And this whole fatty acid tail will remain the same, but this will become this. So this head group attached to this. And that is a glucosylcerebroside, also known as a glucocerebroside. Sometimes we call them, instead of talking about glucosyl, we say the glucocerebroside. So glucosylcerebroside and glucocerebroside, if you do not see this glucosyl, this word right here, still the name, they are both the same thing. So glucocerebroside, aka glucosylcerebroside. So both them are interchangeably used. So the glucocerebroside is not a phospholipid, whereas a sphingomyelin with a phosphate group is a phospholipid, is a sphingolipid, but it's a phospholipid as well. So that's the typical question that we ask from here. For example, if we just ask as a statement that sphingolipids are considered as phospholipid, then you have A, B, C, D. So remember that if the polar, so this is your polar head group, if the polar head group contain a phosphate group, then it is considered as a phospholipid. Again, repeating, the sphingolipids are considered as phospholipid if the polar head group right here contains the phosphate group. Remember that this is the non-polar or the hydrophobic part. This is the polar or the hydrophilic head group. And if it contains phosphate, it's considered as a phospholipid. So this is a structure, the third type. This is cholesterol, and this is a planar molecule which does have a planar four-membered ring and a tail. And this is a rigid steroid ring structure and a non-polar hydrocarbon tail, and they are integral to many membrane structure and many of the hormones, lipid-soluble hormones, synthesized from the cholesterol as the precursor. So very, very important molecule. All right. So after that, we are looking at that if we just take some lipids and put them in an aqueous environment, in the water, how they are going to behave. Now, why we are doing this, the reason for that is that this gives you an idea that how spontaneously lipid bilayer structure can form. So if we just look at an amphiphilic molecule, remember that amphiphilic means that one part of the molecule is hydrophilic, one part is hydrophobic. So if you are looking at this, this is molecule where you have a head group and a tail. But this has just only one fatty acid tail. This is not a lipid of the membrane. So this is not a membrane lipid. Just remember that. So this could be same like a sodium palmitate. So these are usually what you find in the soaps and the detergents. You actually find these kind of molecules, which does have a single fatty acid tail. If you put them in the water, they pack in the water like this and form a micelle. This is the typical structure that they form. So all the fatty acid tails are towards the core, which is the hydrophilic inside, and the outside is the polar head groups, and this is what is called a micelle. On the other hand, what you have that all the typical lipids that we see in the membrane, they have two fatty acid tails, except of course cholesterol is a different class. But phospholipids, sphingomyelins, they all have these two fatty acid tails. When they pack in a little bit different way. And the structure of that is known as the van der Waals envelope. Here, see that with a single fatty acid tail, the van der Waals envelope is somewhat conical, whereas the van der Waals envelope is somewhat cylindrical if you have two tails. And they pack in a little bit different way in the water. And the van der Waals forces also stabilize the hydrophobic core of the membrane along with the hydrophilic interactions. So these are the natural ways. So think about that when you take a beaker and in this beaker, you just put water in there. And now you put a drop of oil, and these oils are the same fats. And so how the oils or the fats will line up? They will all line up with their tails towards the upside right here. So they will not forming the micelle yet. So you gently put a layer of oil on top of that layer of water. So this is your oil. Just like I say, vegetable oil, canola oil, they are this. So basically each of one of these looks like this. So these fatty acid tails are towards the top, which is away from the water. And this head group is polar. So that is in contact with the water. Now what happens? You take this and you shake this one so that all these oil disperse in the water momentarily, at least. They will all form this micelle. And then from the micelle, what will happen that gradually all these different micelle that they have formed throughout the water, they will all come together, float out, and again form this single lipid bilayer. So this is how naturally the lipid actually orients themselves depending upon the aqueous environment. And that's one of the keys that how the membranes actually are so dynamic, that one membrane fuse into the other. The viruses enter into the cell through these membranes. And when these membranes actually fuse with each other, they just do this seamlessly. Just keep in mind that just if you just think about that this oil water, how you can form the micelle and they all come together spontaneously just by their van der Waals interaction. That's basically what it drives this. The same way, membrane will also come together and form an envelope naturally around the cytosol or the cytoplasm by the nature of its amphiphilic property. So that's all your take-home message from that slide. All right. So after that, we are looking at the liposomes. So liposomes are often referred in many experimental based questions. This is nothing but an artificially made membrane structure where you have a phospholipid bilayer, and this layer is artificial membrane, and you can do a lot of experiment with that. And inside, this is an aqueous compartment. So you can think about that this is the cytosol or the cytoplasmic part of the cell. This is the exterior, and you can just do lots of experiment with those as well. All right, so biological monolayers, sometimes we can ask, is there any biological monolayers? Yes, you can find biological monolayers. Say, for example, our phospholipid bilayer, they look like this. So you have these fatty acid tails, two fatty acid tails, and the hydrophilic head groups. On the other hand, what happens in archaea, so there are three domains of life. So the three domains of life, according to the modern division, you have a bacteria, archaea, and eukarya. So bacteria doesn't usually show this. So in the archaea, what you see that particularly the one which are thermophiles, what we mean by that, they thrive in extreme geothermal conditions. Like sometimes they will be in hot water springs, then hot water, these mud ponds, then undersea hydrothermal vents where the water is almost boiling, and they can withstand boiling water temperature and thrive in those conditions as well. And see that what's the unique thing, as they evolved, what they did, their fatty acid tails have joined together to form a monolayer. So it is just a single molecule end to end. So this is a monolayer because this is a single molecule, single molecule, single molecule next to each other. Whereas here you have 1, 2, 1, 2, 1, 2. That's why you have two layers in between. So in thermophiles, in archaea, you find those. And also sometimes you find the monolayers in the LDL particles. So these are the low density lipoprotein particles. And this is how the cholesterol and different things are moved into our body. You all know that when you do the blood work, we call HDL, LDL, HDL high density lipoprotein, which you want to be a little bit higher. You want the LDL to be usually low, definitely below 200. So those are the things that you know. Structure of that phospholipid monolayer. And these are the same phospholipid that you have on the plasma membrane. Same thing. There is nothing different. Same type of either phosphatidylcholine, phosphatidylinositol, sphingomyelin, same sets of phospholipid. And then in the middle part, you have these cholesterol esters. So, and then you have unesterified cholesterols right here. So what are the different things in between the cholesterol esters on those? And we'll talk about that later. An esterified cholesterol is that you have the cholesterol, and if a fatty acid tail is attached to it, we call that they are esterified cholesterol. Think about that. The inside part is the cholesterol plus fatty acid bonded together in the cholesterol esters, not all of them. On the other hand, these are the cholesterol, which does have the same structure like this, which we just saw before. So just remember that right here, if we're just looking at this structure, just give me one sec here. So this is the entire structure. I just wanted to just clarify that when you make a cholesterol ester, that ester is connected onto that. This is not this tail is the part of the cholesterol. So so don't consider that this as a fatty acid. The fatty acids are associated here, and when it is associated here, we call that as a cholesterol ester. Whereas if you have just this part right here, just this part, this is unesterified cholesterol, which is inserted into the membrane that we have seen in that picture. So this is in the membrane, unesterified cholesterol, just the cholesterol plus here cholesterol ester. And these are associated with a protein called ApoB, which helps keep them soluble in the aqueous environment and can be carried through the via blood. And these molecules are released at the plasma membrane, and they will remember that oil mixed with oil, so lipid mixed with lipid. So when these LDL particles are released on the plasma membrane of the target cell, these molecules will dissolve into the lipid. So lipid will dissolve into the lipid, and the material will be endocytosed into the cell. So we are going to see those in a different context. After that, we are looking at lipids can actually move in the bilayer sheet. So there are several types of movement. One is a lateral diffusion. They can move sideways, they can flex, they can rotate themselves, and also they can flip from one side to the other side, say from an exoplasmic phase outside to the cytosolic leaflet phase. They can do this. But this is thermodynamically extremely energy demanding, because if this head group, which is polar and charged and hydrophilic, if it needs to go through this hydrophobic core, it needs a lot of energy. And this is done by a molecule known as a flippase. So flippase is a molecule, an enzyme or molecule, which is a transmembrane molecule. And this transmembrane protein molecule, of course, so this transmembrane protein molecule is the enzyme or the mechanism by which the phospholipids can flip-flop, and that requires ATP or the energy. I have posted one example that someone asked, that why the membrane want to do flip-flops, or what are the reasons. We are going to talk about that in the later part. Sometimes these will be bringing in different carbohydrates. They will be attached to that. And these carbohydrates are needed to be associated with the proteins to make them glycoproteins. So these are the precursor of the glycoproteins, and those are brought inside the cell by flipping these lipids. And one of them is known as a dolicol phosphate. We are going to, not here, we are going to see that dolicol phosphate. And this is a typical lipid which does have the flippase activity. So, and we are going to see that in the unit 2, which does that. So one of the question, very important, and it is a trick question, and you are not going to see it first time in the exam, but it also, even when you read the question many times, it gets you on that question. So I'm telling you that. So see that lipids can move, and all these movement, lateral diffusion, flexion, rotation, flipping, these are all happening with the lipid. And in the question, if we just change the word from lipid to a protein, and we say that none of the answer choices are correct, then a lot of people get confused. So this is how the question is set. Which of the following is incorrect? In a phospholipid bilayer, proteins instead of lipids, just I change one word. Proteins can laterally diffuse, they can flex, they can rotate. Also, they can flip from outside to inside. Then I say all of the answer choices are correct. Everybody picks up all of the answer choices correct, which is obviously wrong because I changed the word lipid to a protein. Proteins do not do this flipping function. Proteins will do a lateral diffusion, sometimes flexion, rotation, but they do not flip. This is a property entirely for the lipid, not protein. So I will just write down, not get frustrated, the important one. So I'm just pointing out that you will see this question in your quiz set. So just be mindful about that. All right. So after that, we are looking at the membrane fluidity, and what happens that all the membrane actually can remodel itself depending upon the temperature. And this is the unique thing. So you just may think is like, what is the context? Like our body is at 37 degrees Celsius. Why this is even necessary, or what is important? This is the most interesting thing, that your peripheral body layers, so that what you have 20, 30 layers of dead skins. Under that, you have all your skin cells that are alive. Then you have connective tissue, adipose tissue, and other tissues. These all have cells. Now, if a person is in the tropics where they're in a constant hot temperature, and there are someone who is near the Arctic region all the time, in Alaska or in the Arctic region, you take these two people, and look at their membrane composition in their skin cells, adipose cells, or in any peripheral tissues. You look at their membrane composition, they will be totally different. Now, a person who is in the tropical area moves into the Arctic Circle, and you check lipid content in their membrane, in their tissues, cellular tissues, after probably a month or so, you will see that their membrane composition have completely changed. So this is a built-in mechanism in the body so that your body doesn't melt or freeze. So that's basically how it is done with all animals, and it has a way more implication than that what we can think about. All of these are based on what is called as a transition temperature. So what we are looking at, say for example, if we are looking at a membrane, just a random membrane here, and this is below a transition temperature. And again, we'll talk about that for each membrane composition, there is a specific transition temperature. Now, remember that when we calculate that for our calculating purposes, we have to just consider a membrane or an artificial membrane that is made up of only single type of phospholipid, because each phospholipid will have a different transition temperature. So for example, if we just think about that we will mention that if you just think about that, if we have a mixture of all these different phospholipids in the membrane, calculation of the transition temperature becomes much more difficult. If they are made up of all phosphatidylethanolamine, all phosphatidylserine, or all phosphatidylcholine, then you can measure the transition temperature much more easily. So for all experimental purposes, the scientists actually build a liposome with only one kind of lipid and study these transition temperatures to have an idea. But in nature, it is much more complex than that. So just think about that I'm just saying that suppose a transition temperature is about 35 degrees Celsius for a particular membrane composition, just random arbitrary number. Do not have to memorize anything. 35 degrees Celsius. Below 35 degrees Celsius, the membrane will look like this. And if you just look at the thickness of the membrane right here, this is pretty thick right here. So I'm just saying right here. On the other hand, what happened if you subject that membrane to a temperature which is more than 35, say for example, about 40 degrees Celsius. See what happens that all these fatty acid tails will start vibrating. And these are the random Brownian motion that you are saying, the molecular vibration, and they will pack to each other and see the thickness of the membrane is reduced from this. So right here, this is a much more narrower membrane compared to that, because some of the fatty acid tails actually gone into this work. And why this has a thicker gel-like consistency, and here you have a fluid-like consistency. And the transition temperature is directly proportional to fatty acid chain length, less, and degree of saturation, more. So just think about that. We have all these different length of the fatty acid chains. Remember that you have two fatty acid chains, 1 and 2. So you can have like 14, 15, 16, 17. On the other hand, 14, 15, 16, 17. These are all the carbon numbers of the fatty acid chains. They are dependent on that how many of them are there. So if you have a 14 carbon length, the transition temperature of that will be lower than a 17 carbon. On the other hand, if there is just only 1 or 2 unsaturation compared to all saturation, the transition temperature will drastically vary. And that's the most important part in here. And we'll spend some time on calculating some of these. So what we are looking here, that these are short fatty acid tails, and some of them have unsaturated tails with the kinks. The transition temperature is less in there, and they are easier to melt, and they are more fluid, like this. On the other hand, these are long fatty acid tail, and they are saturated hydrocarbon tails, and the transition temperature is harder to melt. So from here, that as we said, that if you just in general, overall, if you look at a person in a colder temperature versus in a hotter temperature, so a person in the tropical area will have a composition with long fatty acid tails, harder to melt, Whereas in the Arctic region, just arbitrarily saying, a person will have short fatty acid tail with unsaturated, where it is easier to melt, that in a colder temperature, they like to keep the membrane fluid. This is more important. They like to keep the membrane supple so that it does not freeze. Because does not freeze. That is the easier to melt for sure, but that is not a threat for these people. For this, you want to keep those fluid enough so that the molecules can pass and it does not freeze. Where here, you just want to keep them in the condition that they are a little bit more rigid. You want them rigid to some extent so that they don't melt, which will compromise the integrity of the cell. So outcome of that, short fatty acid tails, which are more fluid, helps the people in the Arctic so that their cells don't freeze. Whereas in the tropical area, the rigidity of the membrane so that they don't easily melt in the environmental temperature. Now, let's take a look at this, and this we'll spend some time on that. And you are going to see a question in the critical thinking question. Look at that question very carefully. You are going to have almost a similar, if not word by word, a similar example of that in your exam as well. So you have to calculate that. You do not need to do any arithmetic or any calculation, just an overview of that. So let's take a look at how we are looking at that, the transition temperature of a what, and I will just walk you through. So the idea is, which property, saturation or length, contributes more to the fluidity and the transition temperature of the membrane. And we looked at that, and we understood that the saturation does have more effect on this compared to the length. So we are going to see that. Let's take a look at the different fatty acid tails. So just again, remember that when we are looking at any of these phospholipids, you are drawing a picture like this. You have a head group, and you have two fatty acid tails. This is your general structure. And if we are looking at, and we will just mention that when we give you then an example of that. So we will say that one of them actually contain a 12 carbon length, another is also a 12 carbon length. So suppose this is one of those, and there is 0 double bond, transition temperature is -1. See that 14, you have added a little bit more from there. You have added a little bit more, 2 hydrocarbons, 23, then 16, 41, 18, 55. So from 12 to 18, you have about 55-degree difference in between. So you had to add 6 carbons onto this to reach about 54 degrees. And so on, say 20, now it is becoming less. 20, then 66, 22, 75, 24, 80. But see that you have an 18 carbon length, right here, 18 carbon, and you just added one unsaturation, and from this 55 degrees, it dropped to almost close to 1 degree. So one unsaturation dropped the temperature 54 degree. So one unsaturation. Whereas here, this is also about, say, 56 degrees difference, and you needed for this 6 carbon to be added to increase that to 55 degrees. So 1 versus 6. That's why we say that saturation has a much more drastic effect compared to the length of the carbon. So see that 18 carbon length, and you have 2 unsaturation, it goes -53. And then 3, it goes to -60. And as we say that usually we do not actually see more than 3 unsaturation in a fatty acid tail. And if there are more unsaturation, the degree of the transition temperature actually is not directly proportional. See that 1 or 2, that is the maximum that you do. That even from 2 to 3, it is about 7 degrees. So the most drastic is just only 1, 2 kinks in the tail will do all the job. So let's take a look at that. Do not memorize these values. Know the concept of degree of unsaturation have more drastic effect on transition temperature than fatty acid chain length increase. Do not forget, phospholipid always have 2 fatty acid. This is 1 and 2. They can be same length or different in terms of carbon chain length and degree of unsaturation. In a same phospholipid molecule, One fatty acid can be 14 carbon length, fully saturated. Another can be 18 carbon length with 2 unsaturation. So what type of question what we are going to ask? So if we're just trying to tell you that you calculate the transition temperature of the 3 different membranes with different composition. Now, we have to tell you that all these membranes that we are giving to you, they are made up of same type of phospholipid. We will tell you that they are made of all phosphatidylcholine or all phosphatidylserine or phosphatidylethylamine. Otherwise, it will not make sense. So suppose you have an A, and suppose, as I said right here, one is 14 carbon right here, one fatty acid, 14 carbon, fully saturated. Another is a little bit longer, 18 carbon, and it has 2 unsaturations. Then you have another one, say B, right here. You have B, and you have 14 carbon, this one, and you have 14 carbon, this one. Both of them are same length. This was 14, 18 with 2, and 14 and 14. And then you have another one right here, which are, one of them is 18. I'm just randomly talking about 18, and this is your C. So this is a type of question that is in your quiz, and this is a type of question that we can ask. So now, can you tell me if I just give you the different transition temperature, can you match them to these? So I'm just trying to see that with this. So say for an example, see that this is your 14 carbon length. So I'm just randomly talking about that suppose the transition temperature for this one is 23 degrees Celsius. Well, the thing is that I'm just picking it up. It is not that it is right here. You do not have to memorize these things. Now, if it is an 18, suppose this is 55. This is your 55. This is the longest one. So that's what it is. But see that what is going to happen. This is 14. If this was 14, it's supposed to be 23. Now this is 18. This is a little bit longer, but it contains 2 unsaturation. See that 2 unsaturations cause what? Like this. So sometimes we can just draw this 2 degrees Celsius. I'm just saying 2 degrees Celsius right here. So if we just randomly have, suppose a 2 degrees Celsius, suppose something in the middle like a 23 and a 55. If we have these 3 numbers and tell you to match those with this, how will you match that? So the first thing that what you will do, that which does have the longest carbon, that will be always the largest number. Then you have a moderate length, that will have probably the middle number. So what you will remember, that whatever composition what you give, look for that which one contains the highest number of unsaturation. That temperature will always be the lowest, whereas then a lower carbon content will have a middle temperature, and a higher carbon content will have a higher temperature. So that's how you just consider those 3 different types. And this is a typical type of question that we are asking. And again, remember that these numbers doesn't mean anything. Do not have to memorize those. Just remember that the number of degree of unsaturation has the most drastic effect compared to the length of the carbon. And based on that, you just can predict which will have a higher or lower transition temperature. Take a look at the critical thinking question, and so whenever you do this, my suggestion will be that when you do this for your exam, you will see this question. Just draw 3 molecules of fatty acid, A, B, and C, and then write the carbon numbers next to that and the saturation. And it will be so much easier rather than juggling in your head that what is what. So again, that will be the easiest way to solve these. All right. So I just spent time because a lot of people actually struggle with these questions. So that's why I just spent a good amount of time in here. All right. So here, sterol content can influence the fluidity of the membrane. And again, not any specific question, but this is a very unique thing that cholesterol make the membrane more fluid or less, which it does both on a temperature dependent fashion. So at physiological temperature, cholesterol decreases the membrane fluidity, but at lower temperature, it makes the membrane more fluid than the phospholipid so that it doesn't freeze. Therefore, it is temperature dependent. So this is a very, very unique molecule and has a huge role in membrane fluidity control. All right. So after that, we are looking at the biological asymmetry of the membrane. And this is the first thing that we talked about. And again, we are repeating that. So when we were looked at the very first slide, we talked about that you need to know the orientation or the topology of the phospholipid in the membrane. So again, from here, that is hydrophilic heads face, exoplasmic face, and cytosolic face. The fatty acid tails are buried in the core. Remember that. So what we mean by the membrane asymmetry. So if you look at the outside of the membrane and the inside of the membrane, they are not made of the same lipid molecules. So here you can see phosphatidylcholine, sphingomyelin, cholesterol. There are some glycolipids. These are lipid anchored proteins right here. On the other hand, what you have that on the inside, what you are seeing that you are looking at this negatively charged phosphatidylserine in a much more, you have phosphatidylethaноlamine, you have phosphatidylinositol, but you are also looking at much more number of phosphatidylserine. But you see that outside, you don't have a phosphatidylinositol right here, this green one, dark green one, because this is a signaling molecule. They are not usually on the outer leaflet. They are in the inner leaflet, which generate this signaling molecule. A phosphatidylinositol or phosphatidylserine that are more in the inner side of the membrane that also make the inner side of the membrane negatively charged. So there is a polarity, there is a polarity difference in between that. So usually most of the membrane are in between, say, minus 55 millivolt to about a minus 75 millivolt in potential difference in between that, because the inside is negatively charged due to the negatively charged, not only the phospholipid, but also the proteins that are present on the inner side, on the cytosolic side, they are all negatively charged. And that's why the inside of the cell is negatively charged compared to the outside. And that has a huge implication in the signal transduction, because sometimes what happens that when certain molecules bind and open some of the gates and some of the molecules like sodium rush in, there is a reverse in polarity from minus 55. It will just go to plus 20 millivolts. So the inside becomes plus, and that actually propagates a signal for an action. We call this an action potential. So again, we are not considering those here, but just remember that the membrane composition are different and there is a membrane asymmetry. All right. So how these membranes are bent, all these curvatures are achieved. So if you look at the van der Waals force of the different phosphate, they are very interesting right here. So you see that this is made up of phosphatidylcholine, PC. So this is 2 phosphatidylcholines. The membrane is 3.5 nanometer. If it is phosphatidylcholine and cholesterol, they become 4 nanometer. If they are made up of sphingomyelin and cholesterol, they become 4.6 to 5.6 nanometer. So just changing the lipid composition, you can change the lipid thickness. And that is a huge implication whether the membrane will melt or freeze easily or not. Then you can see that right here, phosphatidylethaноlamine does have a smaller head group. That's why the van der Waals envelope is to some extent conical. And this is your phosphatidylcholine. And see that how strategically the membrane asymmetry actually provides a roundish structure for membrane structure. So here so you can see the outer leaflet contains the cylinder-shaped phosphatidylcholine, whereas the inner leaflet contains this conical-shaped phosphatidylethaноlamine, and that creates a bend in the membrane. So these micro adjustment, it can be done by moving the lipids into the membrane constantly. And it's fascinating that how these can be done. Say, for example, this is an HIV that is budding out of the cell, or these are the Golgi complexes and all these kinks and all these are all biological membranes. Remember that we are talking about the membrane, plasma membrane mainly, but all biological membranes, which are endoplasmic reticulum, Golgi, and all mitochondria, chloroplast, a nuclear membrane, they're all biological membranes, and they are all the same properties, and they are made up of these same lipids and the same composition everywhere, no matter what. They exchange their parts constantly, and it's a very dynamic structure. All right. So after that, we are looking at the faces of the cellular membrane. And this is one of the things that sometimes people get a little bit confused with this, that what is the exoplasmic phase and what is the cytoplasmic phase? So first, when we are looking at a cell and a couple of organelles, and the organelles are 2 different types. One of them is a single membrane bound, And some of them, like a nucleus and mitochondria, are double membrane bound. And if we are looking at the different structures, let's see that how they are different. What we are seeing, if we just take lipid bilayer from here, plasma membrane, the exoplasmic phase is gray, and the cytoplasmic or the cytosolic phase is orange in color, which is next to the beige color, which denotes the cytosol. All right. Now, if you have a membrane which is bound like a Golgi, lysosome, endoplasmic reticulum, which are bound by a single membrane, the outer face of the outer membrane, that is equivalent to the cytosolic phase. That's why they are orange. Whereas this is the lumen, which is the white part. The luminal part is the gray, which denotes the exoplasmic phase. And why it is so. So just think about that the inner compartment of the Golgi lysosome under the endoplasmic reticulum is equivalent to the exterior of the cell. Because what is happening that this vesicle, say for an example, would arise from the Golgi, will travel and it will fuse with the membrane. So this will fuse with the membrane and it will flip itself outside. So the outside will be flipped outside and this membrane will become continuous. So this orange part, which is the cytosolic part, that will become continuous with this orange part. This orange part, which is the cytosolic part, will become continuous with this part. And the outer part, this part is the luminal side, that will flip towards the outside. That's the reason that the inner topology of the membrane denotes the exterior part of the cellular environment. And this is how the membranes are recycled. Now, the concept becomes a little bit more complicated when you have a double membrane structure. Here, you cannot consider the membrane if you go from this side all the way through. Then you will get totally confused, because what we are seeing that right here is a cytosol. You have the cytosol, so you have this outer leaflet, cytosolic. Then your inner is your luminal side of this in between the membrane space. This is your outside. Then you would think that this should be again your luminal, and how this becomes, because this is outer, inner, outer, and it does not work that way. So what you have to consider, just make sure that when you are considering this membrane, you are only considering it from this side. When you are considering this membrane, you are considering it from this side only. So how it works, this outer part, so the in between the membrane, the white space, that is equivalent to the luminal phase. So the cytosolic part is cytosolic, so that's orange. And then the luminal part is the exterior part, which is gray. Whereas the nucleus, the inside, you consider that this is equivalent, the nucleoplasm is equivalent to the cytosol. So when you are going from here, this outer part is orange, which is a cytosolic phase, and then this gray part is facing the white luminal phase, or the exterior phase. And this is the same thing for the Golgi as well. When you are considering this membrane right here, you are considering it in perspective of this cytosolic phase, then luminal phase. When you are considering this inner membrane, you are considering the right here. This is the matrix of mitochondria, and matrix of the mitochondria, that will be equivalent to the cytosol. So the membrane that is surrounding that, the inner part is cytosolic and the outside is the exoplasmic phase. If you just read the blurb, you will see that I just wrote it down. The video is in very detail, because this is where people get confused. Because you will always, if you just go from one side, you say that this is outer, inner, outer, and that's not the case. That's where, so here, this is your outer membrane, this is your inner membrane, but each of them you consider from different side when you are looking at the leaflet. So it is better to use the term exoplasmic phase and cytosolic phase rather than using inner leaflet or outer leaflet, because for particularly the inner leaflet or the outer leaflet for the nucleus or the mitochondria become very confusing. So to tell that which is the cytosolic phase, which is the exoplasmic phase, is much, much easier. So just spend some time on this one. So here, this is the same thing. And this outer inner just tells you not the face. This is the membrane. This is the membrane, not face. Not face. And just make sure that you correspond those faces with the orange. Orange one is a cytosolic and the gray one as the exoplasmic phase. You will do for both, for the outer and the inner membrane. The difference is that when you consider the outer, you will go from outside to inside. When you do the inner, you will go from inside to outside, considering the matrix and the nuclear plasm as the cytosol. So that's the basic rule. Let's move on to the next part. We already talked about these. Here we are looking at, again, an important picture. Integral or transmembrane, tightly associated. These are peripheral proteins. And whenever you see a protein with a tail, 1 or 2, remember that doesn't matter. If you see that there is a protein which does have 2 tails, that will be a lipid anchor as well. If you just have that whole thing. So this is actually easily missed. And remember that the lipid anchored protein are often, they are glycosylated. So they are glycoproteins. Remember that. They are glycoproteins. And we are going to see a typical type, which is called a GPI, glycophosphatidylinositol. So if you see that there is something written as like GPI and has 2 tails, remember that this will be a lipid anchored protein. This is a question that is highly missed in the exam. So I'm just making sure that you just pointing out those GPI anchored proteins are lipid anchored proteins. All right. So we'll come to the GPI anchored proteins in detail in a second. All right. So we are looking at, again, some of the transmembrane proteins and the typical type of forces that are associated with that. So what we are looking at, that these are as alpha helices. This is a single pass transmembrane protein, multi-pass transmembrane protein. And what are the different forces that keeps these together? So there are hydrophobic and van der Waals interaction. That is actually important as well. Hydrophobic and van der Waals. And see that this part of the protein, which is towards the exterior, is hydrophilic. And the part of the protein which is towards the cytosol, that is hydrophilic. But the core part is hydrophobic, this part. So how this is maintained, remember that there are amino acids which are hydrophilic or polar charged. They tend to be on these outside part, whereas the amino acids which are non-polar or which are the aromatic amino acid, there are many of them, non-polar amino acids, which are hydrophobic. They pack so that they can be happy in the lipid bilayer or the fatty acid hydrophobic layer. So hydrophobic van der Waals interaction. So a typical question is asked, if we just look at a protein in here, what are the different forces that are acting in this part? So say, for an example, right here. So these are stabilized by van der Waals interaction, as well as hydrophobic interaction, which is the same. You do not have to memorize anything from here, just gives you just a single pass alpha helix. Mainly that what are the different domains, like in extracellular cytosolic domain, and this is the intra-hydrophobic membrane spanning domain, and what type of force is stabilizing those, van der Waals and hydrophobic. That's your all take-home message. Then these are just a couple of different proteins, just only for your visual representation. We are not going to ask any questions from those. This is a bacteriorhodopsin glycerol channel. This is a multipass proton pump photosynthetic reaction center. And this is a little bit different compared to those. That's why just I put it in there for perspective, not required for any questions. These are mainly alpha helices. And you have these intra-chain hydrogen bonds which stabilize those. This is also alpha helices. Whereas there is another structure which is actually found, and we're going to talk about this, is a beta barrel structure. Think about that these are like paper towel rolls. And if you push a paper towel roll through the plasma membrane, the structure that you will have, that is called a beta barrel. So what is the difference? The beta barrels are made up of these beta sheets. They are stabilized. by interchain hydrogen bonds rather than intrachain hydrogen bonds in the alpha helices. So these are the beta strands. And aquaporin is one of the most common one. Aquaporin is the water channel through which water can move across the plasma membrane. They are these porin family. So this is a beta barrel porin from E. coli. Then this is important. So here you can see that this is a GPI anchor. And this is also, you can have an acylation or a prenylation, and these are all lipid anchored proteins. So even if you see a protein like this, and these are the carbohydrates, if you see these are all lipid anchored proteins. So other than that, what kind of proteins you can have? If you have a protein like this will be a peripheral protein. If you have a protein like this will be an integral protein. But whenever you see these tails, you know that those are lipid anchored proteins. That is important. And about the visual representation is important. So that's why I've been reiterating it many times. All right. What are the different types of lipid anchor that can happen? So one of them is acylation. Two types of acylation, and one is a prenylation. So what are the difference in between them? Let's take a look at that. So what is happening here? Each one of them is a protein, and the proteins can be anchored on the membrane by help of a fatty acid. And the anchor can attach itself to the N-terminal. It can attach to anywhere in the middle of the protein, or in the C-terminal. That's the basic difference. Now, the second question is that what kind of anchor? Say, for an example, here, this is a myristic acid, myristoyl anchor. So from myristic acid, myristoyl is the adjective of that. So this is a myristic acid, that's the fatty acid. If this is added, this is a myristoyl anchor. This is a palmitic acid, palmitoyl is the adjective of that. If a palmitic acid, fatty acid is attached to an anchor, it will be a palmitic acid anchor. Whereas here, it is a isoprenyl group. It's a farnesyl anchor, which is another, this isoprene unit group. You have this repeated isoprene unit, and this is isoprene-based prenylation. So this is a prenylated. But both of them are acylation, because you're acylated with a fatty acid, acylated with a fatty acid, and this is a prenylated by a farnesyl isoprene unit. So here you have an amide linkage. So remember that from your basic biochemistry, that your amino acid starts from NH2, CH, side R, and then you have a COH. So this is your N-terminal, and this is your middle group, side group, and your C-terminal, it will end with this COOH. And again, this can ionize into NH3 plus, this can ionize into CO minus to form the deuterion. So here, what we are looking at, this is the bond with the N-terminal. It's an amide linkage between the terminal amino group and a myristic acid. So this is basically anchors a protein by anchoring it from the N-terminal of it. So this is your N-terminal anchor. Whereas there are other type of anchors where there is a protein can be anchored wherever there is a cysteine. Wherever there is a cysteine, the cysteines contain the sulfur, and it forms a thioester linkage. So this is an ester linkage with this palmitic acid, and this is a sulfide, a C double bond O with the sulfur, and this is your thioester linkage. And this could be anywhere in the protein, wherever there is a cysteine. It could be anywhere in the entire protein. On the other hand, what happens if the C-terminal right here, C-terminal contains a cysteine, then a thioether linkage binds it with an isoprenyl group, which is the farnesyl anchor. So these are the three different lipid anchored protein: acylated, two acylated, one is prenylated. All right, so different mechanism, that's all you need. So the next one is a GPI anchor. This is extra important. This is a question that is missed by a lot of people. So here, the structure of this whole thing is that if I just draw the membrane, you have a protein. After that, I'm just drawing it in a different color. After protein, and after that, you have a middle portion with carbohydrate, and then you have the last portion. Oh, sorry. So paint a different part with, say, for an example, like this, which is the fatty acid tail. So this top portion is the protein, then you have the carbohydrate, and then you have the lipid. So this is a glycoprotein, this is a glycolipid, everything. So that's why we call it is an GPI or glycophosphatidylinositol. So here you have a protein to which you have an NH2CH2CH2 PO4 right here. This is a phosphoethanolamine right here. And this phosphoethanolamine is connected to three mannose. These are mannose. Remember that? These are your carbohydrate. These are your sugars. So all the different names for the carbohydrates can be used. This could be sugars. This could be polysaccharides. Just remember that all the different names that we represent with that: sugar, polysaccharide, carbohydrates. So, and this is N-acetyl glucosamine. N-acetyl glucosamine, which is just a glucose molecule, and it does have an amine group in the sixth position. Here, you have an amine group in here. The typical structure is that you have a protein with a core carbohydrate, or polysaccharide. Just remember that this is the most important part. They are all covalently bonded. They're all covalently bonded to a phospholipid right here. So where is the phospholipid? You can see that this is your phosphate right here. This is your phosphate PO4, and you have two fatty acid tails. So this structure is like why it becomes complicated. This structure is sideways. Structure is sideways. This is your fatty acid tail. This is your fatty acid tail, which are these two tails. And that's why people get confused with that. So this is your protein core polysaccharide. Then you have the phosphate group, and the fatty acid tails are growing. So the membrane will be on this. Most of them we are drawing like this, but the membrane here is going in this direction, and these fatty acid tails are buried in there. So this is placed sideways in that picture. So just visualize that. That will help. And if we just convert that picture into the proper, like these red thing is the membrane. If we just put it that way, just remember that the red portion is the protein, which is the equivalent of that. The green portion is the core polysaccharide or carbohydrate. This word is so important. A lot of people actually miss this word. Core polysaccharide and the fatty acid all are covalently bonded. So if I just give you a thing that where you have a protein with a core polysaccharide bonded with the phospholipid by covalent bonding, what kind of protein it is or what kind of lipid it is, just remember that it is a lipid anchored protein, which is a GPI anchor, or it is a glycophosphatidylinositol. There is no other protein, carbohydrate, and phospholipid connected this way other than this. So this is a very unique one. Make sure that you remember that. So right here, you can see that blue portion is the protein. This is the core polysaccharide, mannose and the glucose, and this is your phosphate and the fatty acid tails. This is your GPI anchor. So that's very important. All right. So after that, let's see what it is. So these are peripheral proteins. Remember that the peripheral proteins can interact directly with the membrane or with another integral membrane protein that we are seeing. Protein-lipid binding, protein-protein interaction, both can happen. Phospholipase A2 is, I do not think that it's needed much more, just a descriptive part. It degrades the membranes. These generate also signaling molecules. We have seen that. It can cleave like a phospholipase C. There is another protein which cleaves the phosphatidylinositol. So this is just descriptive. Don't worry about it. After that, we are coming into the detergents. So the detergents are help to extract the proteins from the membrane, or essentially for membrane proteins. And these detergents are nothing but that some of these mixture of detergents are present in our household detergents that we use as a soap. So where is the connection? Just remember that when we are washing clothes and everything, or even washing our hands, we are trying to get out the grease and the associated proteins with those. And we have to use something which has an amphipathic or amphiphilic property. So a part of the detergent molecule is essentially hydrophilic, a part of it is hydrophobic. So what happens, the hydrophobic portion binds to the lipid, and then the hydrophilic portion wash it out and takes it out with the water. And this is how all the detergents, soaps work. There are two different types of detergents that we use in membrane biology. One is a denaturing detergent. So the example is sodium deoxycholate and sodium didecyl sulfate, or SDS, very commonly used. Remember those because these are important. So what happens that these ionic detergents do have a plus charge right here, Na plus, and then you have a COO minus. So they have charged groups, Na plus, see right here, SO minus, part charge. These are called ionic detergents because part of them are ionized. Whereas there are other type of detergents which are non-ionic detergents, like say, for example, Triton X100 or octyl beta D-glucopyranoside, or commonly known as octyl glucoside. So here you can see that there is no charge groups, but just remember that how this works. This blue part right here is the hydrophilic part right here. On the other hand, this yellow part is the hydrophobic part. Here, the blue part is hydrophilic. This part is hydrophobic. The same way where you see that hydroxyl, as well as Na plus, CO minus, these are the hydrophilic part. Whereas this portion right here, this planar molecules, deoxycholate, this is essentially cholic acid, which is present in the bile, that what you have for saponification or emulsification of lipid for digestion, extracted from there as well. That's present where sodium deoxycholate. And then sodium didecyl sulfate, also this is your hydrophilic group, hydrophobic group. And remember that this is a CH2 and 11. That means that CH2, this grows all the way to the 11. So there are this is 11 chain length long. So it's a long hydrocarbon tail. So what is the difference in between that? That due to this nature of these, although they are not charged, what happens that when you put them in the water, they form hydrogen bonds. See that right here, you have, these are the, by property, they form these hydrogen bonds, which is a slightly negative and positive charges are formed in here, which is not like these solid charges like Na plus, 1 plus charge, 1 minus charge, which is in there. But that's basically what we called as a polar group, polar but non-charged. Remember that. These are polar but uncharged, where this is of course polar, but these are charged groups. So why there are two different types and what are the two different things that what we use? So one thing that what we do, that's remember that the proteins do have the alpha helix, or they can have the beta sheet. So this is your alpha helix, or they can have these beta sheets. So usually when you use a sodium deoxycholate or sodium didecyl sulfate, and they are stabilized by these hydrogen bonds in between, and then they have these hydrogen bonds in between them that keeps those. When you're using a denaturing ionic detergent, break hydrogen bonds and denature the protein. So this protein will become flat like this. Denature. So that's why we call the denaturing, or denaturing what? Denaturing proteins. So they will go to their linear peptide structure. And if that is the purpose, that you just only want to extract the protein from a membrane, you do not want them to their native structure or native functional structure, you can use this method. Why you use this method? Because these are cheap, and if you have a bulk extraction, this is actually cost effective. That's basically the purpose. On the other hand, if we just look at the Triton X100 octyl glucoside, these are way more expensive and non-denaturing. And what it will do, that they do not denature the proteins. So if you have an alpha helix or a beta sheet, you can get them in their native form. So for example, if you have a membrane protein, which is an enzyme, and you want the enzyme activity to be intact so that after isolation of the protein, it is absolutely functional, then you will use a non-denaturing or non-ionic detergent for functional proteins. Remember that, functional proteins, which will retain their original structure. Now, in some cases, after you denature the proteins, you can refold them back in a refolding buffer. In some of the cases, that is a possibility, but not always that is successful. Whereas this is always a safe process to extract proteins in the functional form or non-denaturing form. The typical question that we ask is that if we have protein that we are extracting from a membrane and you want this protein to be functioning, which of the following will you use to extract the protein in a functional form? So remember that you can use a Triton X100 octyl glucoside, but you cannot use sodium deoxycholate or sodium dodecyl sulfate. So these are some of the important parts in here. So let's remember that. All right. So after that, one thing also is important that let me just go back in here. Right here. All right. So here, this is actually important as well. So something that I should have told. So to remove the peripheral protein, just sodium chloride and salt is more than enough to break the hydrogen bonds, and you can extract then sodium chloride. So peripheral proteins can be extracted just by adding the salt or the sodium chloride. Whereas this is the integral protein. Remember that the integral protein will need detergents. This will need the detergents, which we just talked about. And remember that you can use the detergents in the ionic form that will denature these alpha helices. On the other hand, if you just use it non-ionic detergents, it will just retain that alpha helices and it will extract it from the membrane. But if you just use just regular salt, you just 0.3 molar, 0.2 molar salt solution, that will break these hydrogen bondings of the peripheral proteins, which are very lightly attached, and they will extract those proteins. So this definitely, you will see this question. So we'll see a couple of these pictures and we'll see that how will you extract these proteins. So the green ones, you need salt. How will you extract this gray protein in a functional form? If it's a functional form, you will use, say, octyl glucoside or Triton X100 because they are non-denaturing. And if you ask this like you really do not care, you can just extract them even in denaturing condition, then you can use sodium deoxycholate or sodium dodecyl sulfate or SDS. So these are the type of question that we can ask, and we can use some pictures as well. Remember that. So pictures and extraction, these type of question we can ask. These are just a little bit detail of the Triton X100, where it was just written as is 9.5. This is the length of that, and that is just being shown here, and how this is formed. So this is how an integral protein actually is extracted. So you can see that the detergent molecule actually binds to these, and that's how they are extracted. Nothing specific with that, but this overview of that. All right. So here what we are looking at, that the concept of the critical micelle concentration or CMC of a detergent. This actually tells you too much soap is not good either. So you have to use an optimum concentration of the detergents, depending upon what it is. So for particularly membrane biology, if you want to extract the proteins in a much more purer form, then you use the detergent in below its critical micellar concentration. Because what happens that if you have too much detergent, it actually forms this micelle alongside the proteins. But what happens, some of the lipids are also contaminated in that, and you will not get the protein as purified as you want. But if you can extract the protein with the concentration below the critical micellar concentration, so you can see that this is the protein, only the detergent is bound to that, and it is without any of the lipid and dissolved but not forming micelles. So this is the advantage of that. Use of detergent just below the critical micellar concentration yields transmembrane integral protein in a more purified form without much membrane lipid contamination. This is typical type of question that we can ask. All right. So after that, we are looking at the movement of the proteins in the membrane. So here, what we are looking at, the proteins actually move in the membrane as well. So they do not flip-flop, but they can laterally diffuse and all the time. And the typical picture of this, that is a mouse cell where you have a rhodamine labeled membrane protein. Rhodamine is the fluorophore, and here's a fluorescein labeled, which is also a fluorophore, which is green in color in the human cell. So two different proteins are tagged in here, and you fuse the cells. After you incubate the cells, you see that the proteins actually move in the plasma membrane, and you can actually measure that. So that's basically what you can do. So proteins in this sea of the lipid, they are basically floating and moving around. They are very dynamic. That's basically what it is. Okay, so here the cells can confine proteins in specific domains. And here what happens that these are the epithelial cells, and we talked about this yesterday. So these are polarized cells where you have an apical surface right here, and right here is the basolateral surface. And the apical surface can be lumen, hollow side of the intestine or blood vessel, whereas this is the wall side of the intestine or the blood vessel. So what happens that when these cells, epithelial cells, are connected, there is a tight junctions, which are, we are going to talk about that in junctions in the unit 4 all the way. These tight junctions are areas where there are proteins and cytoskeleton, which we can just think about. They stitch the cells, neighboring cells, to each other. And again, remember that cells are three-dimensional. So this goes around 360 degrees around the cell. The picture only shows two-dimensional nature, but 360 degree, they have these tight junctions. So what happens, any molecule from here, it cannot go through here. It will go back. It cannot go through here. Any molecule that can go from here, it cannot go through here. So everything will come out. There are only very few molecules, like say, for example, we'll know that sodium is one of them that can move through these tight junctions. But most molecule cannot move through these areas. And very interestingly, what happens that the apical surface of this has this green protein, and here you have the red protein, and the cell can find specific proteins in specific domains. We are going to see in the next part that when you go into the transport, that you have particular proteins in here, which are a different type of transporter compared to some of them which are in here. So very different compared to those. So we are going to see that. So depending upon the domain, the cells can decide where to send what kind of protein and confine them there. You will not see the green in the basolateral surface. You will not see the red in the apical surface. So this is just a picture of the tight junctions. Then this is also another very, very important slide. So lipid rafts. So the lipid rafts are called the microdomains on the surface, on the plasma membrane. These are some of the very specialized area of the plasma membrane where there are some specific type of proteins have a high affinity for that. So if you look at the ligands of all of them, you have the unsaturated glycophospholipid all where you have saturated glycophospholipids right here. You can see. Then you have a sphingolipids right here, cholesterol all throughout. But what you are seeing, GPI anchored proteins, acylated proteins, these are prenylated proteins. And along with that, you have these sphingolipids. And of course, cholesterol is all over. If we look at that the first thing that know that which proteins have a high affinity for the lipid rafts, that is your number one. High affinity for lipid raft, that when they are formed, they are sent onto the membrane. Remember that all these membrane proteins we are going to learn in the next chapter. These are all formed in the cell, in the endoplasmic reticulum, modified in the Golgi, loaded onto the vesicles, transported onto the membrane. When these membrane proteins are delivered onto the membrane, they will find these microdomains on the lipid anchors. And one of them, most important, GPI anchored, acylated and prenylated proteins, sphingolipids as well, saturated glycophospholipids. These are some of the proteins which you find in abundance in this area. So this is the domain of the transmembrane protein domains which confine the lipid raft area. So definitely remember that. So a typical question asked that is a little bit different though. So these are the one question can be, which of the following protein have a high affinity for the lipid rafts? That's number one. So the other thing that what you have is that, remember that you have the protein wise, you have GPI anchored protein, acylated protein, prenylated protein. These are the proteins. There are some specific type of lipids, which are the sphingolipids and the glycophospholipids. These are lipids, not proteins. So just make sure that you make a difference between these. These are lipids and These are GPI, acylated, prenylated. These are proteins. So look at the question that what it is asking, whether it is asking that which proteins have a high affinity or which lipids have high affinity. So depending upon that, you will either answer these or you will either answer that. So that's protein, this, lipid, that. That's your number one. The number two is, if we just say that, say for example, which of the following is false? And if we say that if the GPI anchored protein, acylated protein, and prenylated protein move in and out of the lipid rafts very easily, this will be totally wrong. They do not move out, get in. This is the only thing. They are usually with a high affinity, they will just all come in and they will stay there and they will stay there. So they are not move in and out. So if we say that, say for an example, GPI anchored protein, acylated protein, prenylated protein have a high affinity for lipid raft, that's a correct statement. Saturated glycophospholipid, sphingolipid, cholesterol rich, these have high affinity for the lipid rafts. That is true. And then if I say for an example, these GPI anchored proteins or say some of these proteins move in and move out of the lipid rafts seamlessly, that move out, as soon as you see the word move out, that is a red flag that they do not move out. That will be your wrong choice. So this is also another question that is missed by a lot of people. So definitely underline it many times. Very important question. All right. So these are some of the other mechanisms by which the cells can immobilize protein by different mechanisms. And I don't think that there is, these are very self-explanatory. And here what is being shown that how a multimeric assembly is formed. So this is a T cell receptor, and you can see that you have 1, 2, 3, 4, 5, 6, 7, 8 polypeptide chains. So 8 proteins come together to form this T cell receptor. And all of these are separated and transported into the membrane as a transmembrane protein. And then they find each other out and form this multimeric assembly in the membrane. So these are some of the unique things that how the protein dynamic actually works. That also tells that how dynamic the proteins are in their movement in the plasma membrane. No specific question. This is just an overview of that. All right. So module 2, this is a part 2, which is a transport of the small molecules. So we talked about the biological membrane, and now we are looking at that how we move substances through it. So you have a couple of different things. So you have a passive diffusion, then you have a transport mediated, which is a protein dependent. And then you have a facilitated diffusion. Remember that it does not require any energy. A molecule binds and then it just changes conformation and just moves through it. Whereas the active transport will require active energy in the form of ATP. Now, the coupled transport actually require energy, but it required in 2 steps. So we are going to talk about that in that part. One part, it may look like that it's not needing the energy, but to generate that condition, you need an energy in the second step, which is absolutely essential. And bulk movement is just one slide at the very end. Budding fusion, of course, very energy dependent. All right. So after that, we are looking at the different transporters. So the channels actually move molecules or ions the fastest. You can see is 10 to the power 7 to 10 to the power ions, 1 per second. They are fastest. So these are like a floodgates. You open and they rush in and out. And they are always going from the concentration high to low. Whereas these other transporters and uniporters move molecules from only one direction. And if you just look at the thing that what the legends are, that this triangle means the flat portion is the high concentration, the tapered portion is the low concentration. So this is moving from high to low. This is also high to low. Now we have these 2 types, symporter and antiporter, that moves molecules in opposite directions. And we are going to symporter same direction, antiporter opposite directions. And they move about 10 to the power 2 to 10 to the power 4 molecules per second. Whereas the slowest ones are usually the ATP powered pumps, which move molecules against the concentration gradient. See that compared to all of these, that this is opposite. And here, if you carefully look at that here in the symporter, this molecule, red one, is moving along the concentration gradient, whereas this black molecule is moving, or the ion is moving against the concentration gradient. So here, this is moving red from the concentration gradient, high to low. This is against the concentration gradient. So you can think about these as a revolving door that somebody is pushing it and you are piggybacking or just moving in the same direction with that other person without expenditure of any energy, or the opposite directions. Yep. So that's just a relative rate. That's good enough, more than enough. This passive diffusion is through the plasma membrane. Only the small molecule like carbon dioxide, nitrogen, oxygen, they are the only one which can pass directly through the membrane, whereas ethanol is also permeable, but water, urea, these are almost, say slightly, but it's almost impermeable. You need aquaporin or some channel to move that, whereas large uncharged or charged polar molecule, glucose, ions, these are all impermeable mostly, and on which the passive diffusion is dependent. So remember that you will need this to answer some of your quiz question. Depending upon the concentration gradient. So the higher the concentration gradient, so the faster the movement of water will be. So just think about that there is a dam and you have a lock gate for that. And the dam is totally full up to the brim. And at that time, when you open the lock gates, water will gush through much faster if compared to the water level, it was lower. So depending upon the concentration gradient, it will be faster or slower. Size of the molecule, just as it is, if it is a larger molecule, it will diffuse slower. If it is a smaller molecule, diffuse faster. Then the most important part for the membrane particularly is the hydrophobicity. The hydrophobicity is the ability of that molecule to dissolve in the lipid bilayer. And this is mentioned as the partition coefficient. So as the higher the partition coefficient, the hydrophobicity of that molecule actually increase. And what you will see that I will not ask any specific question based on that in the exam, but you will see that there is one question in your quiz, and a lot of people actually have struggled for that. That's why I have answered that question in your discussion post. So go to the discussion post, and it is about the butyric acid and another ion that they are moving and what will be the rate. And I have solved that. Only there are two things that is considered there: the concentration gradient for two different molecules and the hydrophobicity or the partition coefficient of the two molecule, and the size of molecule was not mentioned. So we just mentioned these, that C1, and if the partition is K, then C1 and K1 is equivalent to C2 and K2. And this is how you solve those problems. Very basic, but don't worry about the exam. So here again, so just unipolar, no energy required, high to low concentration, nothing other than that. Here is an important slide that we will use, that this is a unipolar, and this moves glucose molecule. And there are many different types of GLUT transporter. There are actually 14 different mammalian types, 14 different types that is discovered, and they are depending upon they have been different distributions. So GLUT1 is one of the most common one that transport glucose into the most mammalian cells. So what happens that this is a glucose bind to the exterior surface, and then it changes the shape of the molecule and it delivers it inside. So glucose comes from the high concentration outside to the low concentration. But think about this, that when you have a higher blood glucose level after you have a meal, but at certain point, if the glucose is constantly moving inside into the cell, gradually the glucose concentration will increase inside the cell. And eventually the blood glucose level after a couple of hours after your meal will decrease. So in that condition, what will happen technically, that this concentration will become this. And how the cell actually prevent this condition, because the unipolar work both ways. Remember that the unipolar work both ways. That means that they are used to bring glucose in here. But if the concentration is reversed like this, they will move material from inside to the outside. So to prevent this, there is a basic built-in mechanism that how the cell prevents glucose loss from the cell. As soon as the glucose comes into the cell, a phosphate is actually put in there. So it becomes a glucose 6-phosphate. So you have the glucose molecule right here, and you put a PO4, a phosphate in here. And this is your 1, 2, 3, 4, 5, and 6 carbon. So you put a phosphate on the 6th carbon, so which is a glucose 6-phosphate. So glucose is phosphorylated, and phosphorylation of the glucose actually prevents the glucose from leaving the cell. So this is basically a built-in mechanism for preventing a glucose loss. So that's basically the typical question that we ask. So for example, that we have a red blood cell, and exactly that how I described that sometimes blood glucose level goes down after a meal. And what happens to the glucose in the cell? The glucose moves out and all those kind of things, distractors. But the answer will be the glucose remains in the cell because it has been phosphorylated and no longer has affinity for glucose 1 unipolar. Remember that? Just one word actually sometimes throws people off. Glucose 6-phosphate. Any phosphate adding is the process of phosphorylation. Just due to that word, sometimes which phosphorylation, sometimes people miss those questions. Phosphate adding is phosphorylation, whereas if you take a phosphate from a molecule, we call it as a dephosphorylation. So here, the phosphorylation of glucose to make a glucose 6-phosphate is essentially the process. So make sure that you remember that. All right, these are just only some rendition of the molecular structure or the model that how the glucose is moved in here. There are a couple of notes here that the Km concentration and glucose uptake is half the maximal. We will look at some of the related problems in here. All right, so here we will spend a good amount of time in here because we are going to see some questions from here, and you are going to see quiz question as well. So what we are looking at here, we are comparing two different glucose transporter. One is a GLUT1, which is the one that we just talked about right here, GLUT1, with another one, which is a GLUT2 that is present in the liver cell. And if we plot their glucose uptake, what we are seeing that on the x-axis, we have external glucose concentration in millimolar. And right here, this is the initial rate of glucose uptake, and this is in the Michaelis-Menten equation. We consider that as a V0. So this is your V0, initial rate of glucose uptake. So here you can see that passive diffusion means that if you just put a cell in a glucose water, glucose probably is going to move into the cell in vitro experiment. Remember that. This can only happen if you have isolated cell that if you put in a glucose solution. This is not going to happen in your body as such. So that's why I said in vitro experiment. But this is not the main part. The main part are these two. So if you look at the GLUT1 transporter, if you can see that this is the rate of the glucose uptake, whereas a GLUT2 in the liver cell, you have the rate of uptake is like this. And now you can actually calculate the Michaelis-Menten constant for each one of them. And what is the formula? Then you need to remember this formula for sure, 100%. That Km is half the velocity of maximum, half the Vmax. Km is half the Vmax. So if we just look at the velocity, the maximum velocity of this, the half the Vmax, suppose Vmax is 500 right here for both of them. For all glucose transporters, this is the same thing. So this is half the Vmax. So if we just calculate the Km, it's in between 1 and 2. So for this is approximately, if we calculate that, will be about 1.5 millimolar. That's the Km for GLUT1. Whereas if we just look at that, I don't know that where this will go outside, but this when this reaches this Vmax, this should be something like a 20, 25 millimolar, something even beyond that, probably 30 millimolar, 25, 30 outside the scope of the slide. So this is much more gradual. So we can just arbitrarily tell that the GLUT2 in the liver cells, suppose it is about 30 millimolar. I'm just saying that this is just a random number if we just had the entire curve, 30 millimolar, which is very high. Now see that how this affects the transport of the glucose and what is the physiological concentration related factor. These are the two things that happens. Number one is Km value versus transport transport. So that is the first important thing. So if there is a low Km value, that means that the transporter has a high affinity to bind to the substrate, which is the glucose, and transport it. And see that it is just within just like a 5 millimolar, right here, 5 millimolar, just like from the, if you just read the book, it transports glucose at 77% efficiency of the transporter. So very low concentration of glucose, this glucose transporter is really super good in transporting glucose in a low concentration of glucose. Whereas you can see liver cells, GLUT2, this has a high Km, so it has a very low affinity for the transporter, and only in high concentration, say suppose 20, 25, 30 millimolar, it will transport glucose into a significant level. So what does that mean? What it means, just think about that what happens after a meal. If you just look at the glucose level, nothing to do with the carb. We are talking about the glucose level. After the meal, your glucose level goes up, it comes down, and again goes up, comes down, goes up, like down. So this is the cyclic way that what happens. As the glucose level goes up after a meal right here, this transporter transports glucose in a high concentration. So that transports the glucose into the liver cell and store it as a glycogen. And that gradually will drop the glucose in here. But this will help no matter what, say the red blood cells or all other body cells have the GLUT1 transporter. So even in between the meals, now see this very carefully, even in between the meals, this is the area where the blood glucose level is low due to the low Km of the GLUT1 transporter, it can still transport glucose with high efficiency because it has a high affinity of the glucose. So it binds to the glucose very tightly and it finds glucose in very diluted in the blood. Whereas this one only will transport glucose into the liver when the concentration is too high, and then it will stop because you want the blood glucose level to remain at 5 millimolar in between the meals, and after meal it goes up to 20 millimolar. So in this concentration, the GLUT2 works perfectly and drops it down to 5 millimolar and allows the remaining glucose to be there so that it can supply the body with the glucose. And if it is needed, if it even goes even below 5 millimolar, then the liver cell actually break down the glycogen and release the glucose into the blood so that the body can actually consume that. So this is the concept. So what you are going to get from here, the lower the Km value is the higher the transport rate. Higher the transport rate, the higher the Km value, it's a low affinity and it's lower the transport rate. Lower transport rate. So remember that. So here we are typically asked exam related questions. Suppose, for example, we give you something like carb like this, that you have a carb like this. And say, for example, right here, suppose we are saying that we have a glucose transporter that you have, and you have engineered a glucose transporter that has a different Km. So just saying that, say, for an example, you have a wild type glucose transporter that you are working with, and the Km for that wild type glucose transporter, say, for an example, is 2 millimolar. It's a wild type glucose. It's a T2 millimolar. It's Km. Now, what we're saying that when we make a wild type so this is the wild type. So this is the wild type, which means that this is the natural occurring. Wild type means that in the genetics language, that this is the normally available. And then you have an engineer that you modified and you try to develop better, but unfortunately it did not work. And you see that the transport of the engineer actually got down 3 times. So it got 3 times lower. So Earlier, the wild type was transporting it much faster. Now it got 3 times lower. So the wild type, you have 2 millimolar is the Km. Now, if you're saying the engineered one has a 3 times lower ability to transport glucose compared to the wild type, then what will be the Km of the engineered ones? It's much easier than you can think. So all you have to do is you multiply this 2 by this 3, this will be the 6 millimolar. That's the only answer. It is super, super easy. And I'm telling you this question, you will see this question the simple way. It's way more simple than that. So because see that it is proportional to the lower the Km, higher the affinity, higher the Km, higher the affinity. So if we say the rate is 3 times lower, then the Km will increase just 3 times. On the other hand, say for example, if we just give this value as a 6 millimolar, wild type 6 millimolar, and now we say the engineered one is a 3 times higher ability to do that, then what will be the Km of the wild type? 3 times higher. All you will do is like you 6 millimolar, you will multiply with 3, you will have 2 millimolar. Make sense? That if you just increase the speed, lower means it is transport rate is less. This means the transport rate is low. If you just get these numbers, if you just if you have something that is given to you that this is the original and the wild type is something which is lower. This is lower. This was higher and 2 millimolar. And how much lower? 3 times lower. So 3 times lower, I just multiplied the Km by 3 to get this 6 millimolar. On the other hand, if I give you this, that this engineered one was a 6 millimolar, and now the wild type is 3 times faster than that. So that is this. So what you will do that you will the Km will be lower than that. So you will divide the 6 by 3 to find the 2. So just in one, we are giving you this graph to find this. Another, I'm giving you this graph to find that. One is you multiply the 2 into 3 to make the 6. Other, you divide the 6 by the 3 to find 2. So when you see this question in the exam, do not overthink. It is more than it is just multiplying 2 numbers or dividing 2 numbers. Just see that the lower value, that with the reference, what we are talking about, that the new one that you have, whether you have that faster, if it is a faster, the Km will be lower. That means that you have to divide that number with the fold of increase you have. On the other hand, if you see the number is, the rate has become faster, then you have to divide that number. If the rate becomes slower, you have to multiply that number by the times. That's it. On the other hand, you have a little bit more complicated question in your quiz where you have to use a equation. This will not be in your exam, where you have to use the Michaelis-Menten equation, which is V0 is equal to your V max 1 plus Km over C. So I have posted that as well, 1 plus Km over C. So this is what you will see, that there are 2 transporters. One is a glucose and I think another is a galactose. And what you will see that it is given, you have their Km value, that and 1 plus Km over C. This is the concentration at which it is taking place. You have this value, and the Vmax, it is, you just consider that as 100 for both of them. So the equation will become V0 equals 100 over 1 plus whatever value for glucose Km you put there, and the concentration at which the transport is taking place, you put there, and you find a number, is something like a 77. You calculate that for the galactose as well. V0 equals to 100 over 1 plus Km for the galactose, and the concentration at which it is taking place. You put that number, you will see something like a 14, or I don't remember, 1420 or something. And you just find the ratio of these 2 numbers, and that will give you the answer of about 5.5 fold. So just take a look at the equation. So the equation is this. So again, I'm just writing for clarity. V0 is equal to Vmax over 1 plus Km over C. So this is the equation that you need to solve that problem. But this is just only for your quiz purpose. In the practice quiz, you will see that. You will not need that for your exam. For exam, this is all you need, that Km equal to half the Vmax and the degree of the affinity. That lower Km, higher speed, higher Km, lower speed. That's all you need. All right. So, yep. So that will do it. This was probably the detailed part of it, a little bit of math. Facilitated diffusion, water by aquaporin. These are all descriptive parts. There are lower of aquaporin and cellular permeability. Here you have some oocytes, which doesn't express any aquaporin or anything. All these oocytes, they are left in the water, natural environment, and they do not burst. And how they are, because there is no aquaporin on their plasma membrane. But in this experiment, if you inject the aquaporin mRNA and they express the aquaporin, they will gradually burst. So that's the experiment. Then we are going into the transport, active transport, which will require energy ATP hydrolysis. There are many different classes of pumps, and we are only going to talk about just two of them in our purpose. So one is what is called a P plus pump. They have an alpha beta subunit, work with an ATP hydrolysis. Then we are going to talk about these ATP binding cassette pumps, or ABC superfamily of transporter, which works this like a seesaw for transporting large molecules. On the other hand, we do not talk about the V or the F class pumps. So what are these? So the V class pumps are actually work in plant vacuoles. So for acidification, see that they are actually add protons to the plant vacuoles to make them acidic. Whereas the F plus pump is an F1 ATPase. So this is an ATP. It is not a pump, but this is an ATP synthesizing machinery. In the mitochondria, the protons actually fall from the intercellular compartment, from the in between the outer and inner membrane, into the matrix. As the proton is falling down, it moves this pump to bind ADP and PI to make ATP. So this is an F1 ATPase. This is an ATP making pump. So that's beyond the scope of this. So we're only talking about P class pump and ATP binding cassette pumps. So these are an asymmetric pumps. So sodium potassium ATPase, which transports 3 potassium outside and brings 2 potassium inside. In the cell, you always have higher concentration of sodium outside the cell and low concentration of the sodium inside the cell. Whereas for the potassium, you have a higher concentration of potassium in the cell and lower outside the cell. And this gradient is maintained by these sodium potassium pumps. That's an integral part of many physiological processes. So this is just shows you just how it goes, that step by step, high affinity binding, then the ATP side is phosphorylated. Once this phosphorylation happens, these are moved outside and released. And in this condition, the sodium potassium binds and it is brought in. Once it's brought in, it is released and the conformation change is again ready to bind the sodium. So these are all super simple, really do not have anything specific. So this you do not need, but this is your take home. Just remember that if you look at, you do not have to memorize the numbers. But the potassium inside the cell, it is 400 millimolar, and outside in the blood is 20 millimolar. Inside is more, less outside. Exactly the opposite. Inside the cell, it's 50 millimolar. Outside in the blood is 440, less inside. And this is an universal rule. And there are these sodium potassium pumps and other channels which actually maintain this equilibrium. So that's very important. Then we are looking at these ABC superfamily of the transporter. And one of them, which is worth mentioning, is known as a multi-drug resistant transporter, or MDR1. Now, what is the function of these MDRs? We have hundreds and thousands of these molecules in the environment, which are potential toxins. And these toxins can get into the cell, that environmental pollutants and the toxins. And these ABC superfamily of the transporters of the MDR1, they can bind to a wide array of molecules to transport them. So this is the uniqueness. Say, for example, sodium potassium pump will not transport anything other than sodium and potassium. Doesn't have any flexibility. Glucose transporters will not transport anything other than glucose. Very specific. Whereas the MDRs are not very specific. They can actually work with many different molecules to transport many different xenobiotics and environmental pollutants if they enter into the cell. This is a safety mechanism built into the cell. But unfortunately, what happens, these MDRs also are implicated in cancer. A normal cancer cell, have 10 of these MDRs on its surface, 10 of those. And you have these hydrophobic nonpolar drug and toxin, they are entering into the cell. And these MDRs, which is the ABCB1, they are pumping it out. So there are 10 of them, 10 of these pumps in here. You are just putting thousands of these molecules. So you modulate the drug amount so that even if there are 10 pumps working in there still will be enough amount of drug in the cell to treat that cancer. But sometimes what happens, that what we call as a resistant cancer cell, not resistant, sometimes there is an overexpression of the transporter. So this gene duplicates, it undergoes until a promoter. And instead of, say, 10 of those on the plasma membrane, now you have 100 of those. This is just depicted as a much more number, 100 of those MDRs on those. So now what will happen, that once the drugs get into the cell, the cancer treating drug, it will be transported 10 times faster than a native cancer cell. And this is what we call as resistant to a chemotherapeutic agent or a resistant cancer cell. And this is just by increasing the number of those. Why this mutation is happening? This is totally random. First of all, for getting the cancer, many things need to go wrong. You need the mutations so you lose the cell cycle control. And this is one of the other way that enhances the survivability of the cancer cells, where there's a mutation of the MDR. And this is random. This is unfortunate event actually happens where it becomes a resistant cancer cell. But again, remember that the normal function is not for pumping cancer drugs. The normal function of the MDRs to export environmental toxin, which can transport cancer chemotherapeutic agents as well. All right, so these are ways that how they can bind. So these hydrophilic groove, the binding can be direct into the hydrophobic group, or they can dissolve into the lipid, but these details are not necessary. This is also experiment which shows the ABCB transporter is a flipase, which can flip molecules from this outside to the inside. And this flipase molecule activity is shown in this experiment. Again, you can just read the experiment from your textbook, but it's not essential for your exam. So I'm just skipping that part. Many different selected ABC proteins. You can see that these are MDR1s, MDR2s. These are exports lipophilic drugs, exports phospholipid into the bile, and many other functions. The overview of that, again, detail is not necessary. All right. So after that, we are looking at the coupled transport. So what happens in a symporter? Both of the molecules are moving in the same direction. One is moving down the concentration gradient and one is moving up the concentration gradient. Classic example is a sodium and glucose. Sodium goes down the concentration gradient, and then glucose is actually pulled down. So even if the glucose level becomes really, really low at the very end of a meal, so you have digested the meal, it's moving through your intestine, and gradually the glucose level is going down. And even in that condition, there will be very efficient glucose transport because your body doesn't want to transport every molecule that is present in there. So this can happen. On the other hand, this is an antiporter. Sodium is going down the concentration gradient. Remember that sodium concentration is always high outside and low inside. And as it happens, another molecule can move outside. Say, for example, calcium. Calcium is one of the molecules that is moved from inside the cell to the outside because you want to maintain the calcium concentration low inside the cell. So this can happen. So now, when you are having this first part of the transport, you think that there is no requirement of energy, and that is true. But the overall consequence or the process needs energy. So think about that you are suppose you have an overhead water tank and you have put a dye in there and you just put water in that overhead tank. And then when you turn on the faucet, the faucet, the dye and the water actually moves down the same like a simporter. The water brings the dye through the faucet. Now, who put the water up in that overhead tank? You need a pump which needs an electrical energy, solar energy, or gas energy, some kind of energy that you invested in there to put that water up in there. And you need this for that. Who made that sodium gradient? So if you just go all the way back in here, right here, the sodium potassium pump, all the sodium that is coming in through the symport or antiport, the sodium potassium pump is essentially taking these 3 sodium outside to maintain the sodium gradient and also the potassium gradient for other purposes. The sodium gradient is maintained. So this is where the energy is spent for the active transport. So we call this as a secondary active transport because here this is your primary transport, which is a coupled transport, symport or antiport. But due to the consequence to maintain the sodium gradient, you need an active transport, which is called a secondary active transport. So it requires the energy, not directly. What I will highly suggest that for this, you see the supplementary videos. So there are some really nice supplementary videos. These are YouTube videos. These are YouTube supplementary videos. So which actually shows these two very clearly with the sodium potassium pump as well. So take a look at that, will be really helpful. So after that, what we are looking at is that some antiport. This is specifically for the cardiac muscle cell, but this phenomenon that these calcium, sodium, or sodium, potassium, this happens in all different cells have these phenomenon. But this is where there are some serious consequences that can happen. So cardiac muscle cell actually need calcium for contraction. That is a signaling molecule. Remember that we are going to learn later, but calcium is a signaling molecule. It's absolutely required for the contraction. But what is the important part that if the cell has too much of calcium inside the cell, that can do some mis-signaling. So some aberrant signaling, which the cell doesn't want. It wants the calcium, but it wants in a very low amount. So constantly what happens that the calcium is pumped out of the cell. It is pumped out of the cell as the sodium goes down the concentration gradient. So this is a calcium. I will just write down. It's a calcium Ca2 plus, calcium Na plus antiport. Remember that. This is an antiport mechanism because they are going in opposite direction. See, the sodium is going down the concentration gradient because sodium is always outside a higher concentration. And as it goes down the concentration gradient, calcium is moving outside against the concentration gradient from low to high. And this actually keeps the low calcium concentration in the cell. And this is required. You need calcium, but you need in a low concentration. Now, how this gradient is maintained? The gradient is maintained by the P-class pump, which is nothing but the sodium potassium ATPase or the sodium potassium pump, whatever you call it, sodium potassium ATPase, aka sodium potassium pump or P-class pump, all the same. So all the sodium that is coming in, that is pumped out, three sodiums, and potassium is coming in because you want higher concentration of potassium inside. But the sodium is more important. Any sodium coming in, that is getting out. That is maintaining this gradient that sodium is always, due to this transport, coupled transport, sodium is always high outside and low inside. So it can constantly drive that antiport or push this revolving door as calcium goes out of the cell. And this is a normal condition that happens. Now, in some cases, what happens that the cell accidentally transport too much calcium outside the cell, too much calcium outside the limb. And that can make the cardiac cell go into an arrest, which is a congestive heart failure. So there is no calcium. So this is like overexcited cell. It's just trying to pump out all the calcium, but it just overdo the thing. And this is just like a normal phenomenon that can happen. It's a physiological phenomenon, and that can happen. And without this calcium, the cardiac muscle will stop pumping, and that is a congestive heart failure. In this condition, what are the treatments, what we do in this condition? So what we do, usually use a digoxin or the oabin. These are the drugs. When they are injected, what they do, the drug is an inhibitor of the sodium-potassium pump. Sodium-potassium pump. It inhibits the sodium and potassium pump. So think about that when it is inhibiting this sodium-potassium, this sodium is not getting out of the cell. So the sodium concentration is increasing inside the cell. Sodium concentration is increasing the cell. But to pump out all the calcium, what you need is that essentially you need this gradient, but without the sodium pumped out, this gradient will become cylinder shaped like this. That means the gradient outside will become similar to the gradient inside. And this sodium will not move in anymore, and that will not pump out the calcium again. So this will be stopped as well. So this is an indirect effect where the secondary transport, which is the sodium-potassium pump, is inhibited by the digoxin and the oabin, which leads to the intracellular sodium increase. That leads to stoppage of the calcium, sodium-calcium antiporter, which stops calcium export from the cell, and it increases the intracellular calcium so that the heart can function back again. So this is the typical question. Typical one-liners out of take-home one-line question, if I just have to ask, is how does inhibition of the sodium-potassium ATPase increase the force of heart muscle contraction? Just remember, just one line answer is it increases the cytosolic sodium and therefore decreases the calcium export. Increases this, decreases the calcium export. So intracellular calcium level goes up. That's how it can increase power of the heart muscle contraction. So that's basically take home from this. That's it. All right. So after that, we are looking at some of the other transporters. So here, what we are looking at a slice of the intestine. So if we just look at the lumen of the intestine, and here we are looking at a cell which is on the side right here. This is basically what we are looking at. So this side is lumen side. This is the luminal side in here. And this is the blood side of it. And this is a classic cell which has tight junctions right here. So you have the apical surface right here. This is the apical surface, and this is your basolateral surface, and you are seeing the cell sitting sideways. So what are the different type of transporters are there? And you need to remember those. So number one, so all of them, so I will just take this, all of them. So all of them are red labeled. On the apical surface, you have a sodium glucose symporter. They are on the apical surface. So remember that. So as sodium is going down the concentration gradient, glucose is moving in here. Now, some of the glucose, when it's moving in, can be utilized by this cell. But most of the glucose, they are not. See, remember that this is not a GLUT1 transporter that after transport will convert into the glucose 6-phosphate. The huge amount of glucose is moving through the cell, and this is just like a temporary passing area of the glucose, and most of the glucose is moved from the cell because, like say, billions of molecules of glucose, the cell probably needs just a couple of hundred molecules, and the rest for its own function to survive, and the rest is going to transport as glucose. On the basolateral surface, what you have, so you have this here, basolateral surface, you have GLUT2 transporter. Remember that this GLUT2 transporter is also present on the liver. This has a high Km, and how it works, because the concentration of the glucose as it's moving in is high in here. This works in a high concentration of glucose, and it moves glucose into the blood. Now, what is happening that this sodium, as you can see, the sodium actually moved in with the glucose, but you have to maintain the sodium gradient outside the cell. Outside the cell should be more compared to inside the cell. So right here, your number 3 is the sodium potassium pump, or the sodium potassium ATPase is moving the sodium outside. And so the sodium is always outside more, and sodium can move through this area as well. But sodium is, whatever sodium came in is going out to maintain the gradient. And here there is another one, which I did not mention here, but here is the potassium channel. So what happens, these potassium channels are known as the leak channels also. The cell constantly have a tendency to leak calcium from inside the cell to the outside. And this helps in maintaining the cells inside negative charge inside the cell. And all the leaked potassium that is leaving, the sodium potassium pump actually brings back those potassium inside the cell while pumping the sodium out. So if you just may think that why this potassium channel is in here and what exactly the function. So think about this, that this sodium plus charge is coming into the cell and you're bringing 2 plus charges. See that inside the cell is negatively charged, and as soon as this plus charge comes in, the inside the cell will start becoming plus, and the cell doesn't want that. So as the sodium is coming in, it is moving the potassium out so that plus charge is coming in, plus charge is leaving. This is a fast mechanism, because remember that channels are faster, so coming in and the electroneutrality is, or the differences is maintained. Now, sodium and potassium pump works much slower. So eventually the sodium is pumped out and the potassium is brought back. But there is a delay in between the sodium coming in and in between that minus charge can be increased. And to reduce that, the equal amount of potassium is leaked out of the cell to maintain or keep the inside negative. So the details are not necessary, that only the 4 different transporters, where is what, is important. So we can ask you a fill in the blank type of question. Say, for example, that one example of the transepithelial transport in the system, in the intestinal epithelial cells, that includes sodium glucose symporter in the intestine facing membrane. Then you have an energy requiring sodium potassium pump facing the blood side, and a GLUT2 transporter allowing the facilitated diffusion of those outside the cell. So remember that these GLUT2, this is facilitated diffusion. So you are connecting many things in here. Facilitated diffusion, this is a symporter, this is a sodium potassium pump. So these are all the different things are in here. So these are the main things. So again, this one is on the apical surface, which is the intestine. Then you have the sodium potassium, which is on the blood side. This is the blood side. That's why this, remember that, blood side, intestinal lumen. And you have the GLUT2 transporter, which is a facilitated diffusion. That's it. So that's the typical type of question that we asked. Which side has which of the important transporter in this transepithelial transport? So this one is acidification. We essentially ask you, so again, here what you are seeing, that the carbon dioxide from the blood side is coming into the cell, and carbonic anhydrides make the bicarbonate ion, which is with the antiporter brings in chloride into the cell, which goes through a chloride channel into the stomach lumen. So it is becoming negatively charged. So how you manage this negative charge? So what you are doing with that, first of all, the potassium channels right here, K+, they are neutralizing this K+ and the Cl- to neutralize that charge. But eventually what happens, that carbon dioxide also dissolve in the water to make this H+ ion, where you have an H+, K+ ATPase. So this is a proton potassium ATPase. We talked about the sodium potassium ATPases. This is a proton potassium ATPases, which pumps proton or H+ into the lumen and brings the K plus inside. See that the Cl minus make it negative. You don't want it to be too much negative. So K plus manage that to neutralize that. Now, the proton pump is putting the H plus in here and bringing back the K plus. Now see that what happens, how the acidification happens. What is HCl? It's H plus Cl minus. So that's basically the stomach lumen is the hydrochloric acid. That's the acidification of the stomach. So that's the overall mechanism. A take-home message. Which transporter is not present in here? So you see that this is an antiporter. This is a sodium-potassium ATPase or pump. This is a potassium channel. This is a chloride channel. So if we just give you a couple of things that, say, for an example, which of the following is not there? Antiporters, no, it is there. Pumps, no, it is there. Channels, no, it is there. So the only thing that is not there is a symporter. So you had a symporter in your intestine, whereas a symporter is absent in here. So if you remember that, you should be able to answer that question. So do not miss this. Make me proud. So very short period of time, huge content, and that's why you guys need a little bit of extra help. So these are just some of the interesting things that when the channels goes bad, what it is, you do not have to memorize any of those. The reason I only put that many of our families, friends, they have many of these disease, and we just always want to know that what are the underlying molecular mechanism of that. That's why I just put those, and you can just easily understand that how these channel deficiencies, that all these channels or the transporter deficiencies can cause so many debilitating diseases. So the last part, which is the important part, is the cystic fibrosis. What happens in the cystic fibrosis? We are going to talk about this all throughout in much, much more detail. So this was Francis Collins and Laptis who identified the gene in 1989. And there is ABCB7, which is known as a cystic fibrosis transmembrane conductance regulator, or CFTR. And they are present in everywhere. But how this actually works and what is the relationship with the cystic fibrosis? So this is a CFTR ABC pump, but CFTR is a channel, not a pump. This is the most important part, and a lot of people actually get confused that. Remember that ABC binding cassette, most of them are energy consuming. This is one of the exception, and we'll see that. Let's take a look at that, how this works. So the ABC pump has two domains. You have a transmembrane domain here, transmembrane domain 2. Then you have an ATP binding domain 1, ATP binding domain. You have a regulatory domain, which is phosphorylated or non-phosphorylated. When this regulatory domain gets a signal, and we will learn that how the signal transduction happens in later part, that phosphorylation of the regulatory domain allows an ATP to bind to one of the sites. This is one of the ATP binding sites. And then another ATP binds to the next site. At this time, the channel actually opens. So as long as these two ATP is bound to this ATP binding domains, the channel is domain open, and the chloride is moving down the concentration gradient and not up. So why this is a channel and not a pump? Number one, chloride moves down the gradient. That's number one. Pump, it will be against the gradient. Number two, the pump requires ATP. That this pump, typical pump, that every cycle, when you saw that, every cycle will require an ATP. So that means that every ion molecule that is being moved, you will require ATP. Here it is not. You just only need two ATPs to open it. So two ATPs to open it, and it will remain open as long as the signal is there, and it will remain bound to it. And once the signal is gone, this ATP will be hydrolyzed and the pump will stop. So I will write constant ATP supply. ATP is not needed. And I will just write 2 ATP just to initiate. Just to initiate. That's it. So once you initiate the channel or open the channel, it will remain open unless this signal is gone. So that is the difference between the pump, that when you saw that the potassium or the sodium pump, each 3 sodium that you transport and you bring in 2 potassium, each of the cycle will require an ATP hydrolysis. So each of the cycle. Here it is not each of the cycle. It is just only 2 to initiate and open the gate, and that's it. And also along with that, chloride moves down the concentration gradient, and that's why the CFTR channel is not a pump. One of the questions that we ask is the little bit complicated way that, say, for example, that which of the following is true about the cystic fibrosis transporter protein. Say, for example, that if we just say like this, the normal CFTR channel protein is a chloride uniporter, but the CF patients have a mutation that makes it requires ATP. So this is like, it doesn't make sense. So first of all, it is not an uniporter. It's an ABCB pump, ABCB transporter, which is not an uniporter, different thing. But again, if there's a mutation, it requires ATP. No, it requires ATP to initiate no matter what. So on the other hand, if we say that CFTR mutations result in a mutated protein which pumps too many chloride ions out of the cell, it never pumps too many chloride ions out of the cell. So that's not incorrect. So CFTR is a member of ABC superfamily of members of pumps, which pumps chloride into the cell. As soon as you see the word pump, avoid that, because you know that that's a bait. Don't bite on that, because that will be the wrong. It is not a pump. So those are the things. And then we say that CFTR mutation result in a mutated protein which blocks sodium ion movement from inside to the cell to the outside. It has nothing to do with sodium or calcium or potassium. Just remember that it is a chloride and it is moving down the concentration gradient with just 2 ATP bound to its cassette, nothing else. Anything those. And if there are some of these where all of the answer choices could be incorrect, feel free to choose none of them are correct. There will be some answer choices where you have none of them are correct. Those are the sometimes difficult one, but you will definitely see them. So here we are looking at the lining of the airway epithelium and where CFTR is. So here is the CFTR, and what it does that it moves chloride into the airway lumen. And as chloride moves in, I already told that this is becoming negatively charged. So to maintain the electroneutrality, sodium from the blood side is pulled into the lumen. So it becomes Na plus Cl minus sodium chloride. So think about that. Sodium is always outside the cell, and it can move through these tight junctions. This is one of those rare exceptions. So it can pull in. So Na plus Cl minus. Remember that salt always pulls water. So sodium chloride, Na plus Cl minus, that will pull the water through the cell. And this will go through the aquaporin here, then aquaporin here. Both sides will be aquaporin. So what are the different transporter? Water is going through the transcellular transport. So if we just look at the different terms, this is transcellular. It is across the cell. Whereas sodium is going a paracellular transport in between the cell. So once sodium chloride goes in, the water actually goes in and it loses up this mucus and a normal mucus, normal cell lining, hydrated mucus, and that is the normal condition. But what happens that in a cystic fibrosis patient where the CFTR is not working, the channel does not work. That means that no sodium is pulled in, no sodium chloride, no water is pulled in. So the mucus becomes really super thick, and that becomes very difficult for breathing, and sometimes infections can happen as well. So that's important. So just remember that whenever you are looking at the questions regarding the CFTR, it is never a pump, and it always moves chloride down the concentration gradient, never up, and it only transports chloride ion, nothing else. So that's the important one. And then these are very straightforward questions, just one line. Say, for example, that how a cell engulfs or a bacteria or a large particle. That will be phagocytosis. How the cell pick up a fluid. That's pinocytosis, also known as fluid phase endocytosis. And secretion is exocytosis. And these are mechanisms of this. So that's pretty much all. And we will, so this is just the tip of the iceberg from here. Here we are just learning all these processes. And in the next part, we will learn that how these trafficking happens. In just remember the basic definition of phagocytosis, pinocytosis, exocytosis, and that's all pretty much. All right. And if you have any question, just shoot me an email through Canvas. Good luck with the exam.